Today, it's my pleasure to introduce today's speakers, Professor Jesper Andersen, Professor of Translational Hepatology at the University of Copenhagen, and head of the Andersen Group at the Biotech Research and Innovation Center within the Department of Health and Medical Sciences. Professor Andersen leads translational research focusing on immunogenomics, metabolism, and epigenomic remodeling, with the goal of understanding molecular mechanisms that drive liver and biliary diseases and translating these findings into clinical insights. Today, he will present a seminar entitled Immunogenomics and Chemotherapy Resistance in Biliary Tract Cancers. Please, Professor Andersen, you can start your presentation. Thank you so much for being with us today.
Thank you very much for the introduction. I would like to first thank the Fondazione Italiano Fegato for the invitation to speak to you today here at the Yellow webinars, and of course all of you for attending or listening to this presentation. As said, my name is Jesper Andersen, and I'm Professor of Translational Hepatology and also the head of the Andersen Group here at Biotech Research and Innovation Center at the University of Copenhagen in Denmark. And for the next 45 minutes or so, I will discuss with you the current state of immunogenomics and chemotherapy resistance in biliary tract cancers, and it will be a scientific take on this. I'm not a medical doctor, as disclosure. So what we are looking at here is the representation of the myth of Prometheus, the titan who gave humans the ability to make fire. Now the full gods did not like this gift to the humans, and Zeus condemned Prometheus to die an agonizing death every night, represented by the hawk pecking at his liver, at Prometheus's liver. Only for the liver to continuously regenerate and Prometheus regaining life every morning. So in a continuous cycle, dying and living and dying, repeating for all eternity. Now, if this story was anywhere close to truth, of course, why are we then today discussing cancers in the liver and the bile duct system? The liver should then in itself regenerate the initial damage or the insult that has happened, causing this progression that leads eventually to cancer that we will be discussing today. However, when that is said, it is of course still very impressive that the liver that is facing a lot of insults every day from what we eat, how we live, and so forth, that we are not all actually facing liver cancers, and it indeed is the major organ in cleaning up after how we are living, and does continuously regenerate based on those insults. When that is said, for this talk, I have no particular disclosures. I would like to initiate by saying thank you on behalf of all the authors of the papers that I'm going to discuss with you today, to the patients, the families, and the care medical staff that has contributed to the data that we are using for these research projects within the contributing institutions, as well as all our excellent collaborators around the world. And of course also include a thank you to the members of my laboratory who performed the studies that I will discuss. This is the outline of the talk. I will give you a short, very short background. We can discuss it afterwards on the current state of genomic-based patient stratification and targetability of biliary tract cancers. Then there will be two main scientific parts to the talk: the genomic stratification of intrahepatic cholangiocarcinoma, our take on that, and the perturbation mechanisms that we have discovered that leads to novel therapeutic opportunities. And then towards the end, or the last part of the talk, discuss the idea of precision chemotherapy. So actually making what is typically overall imprecise more precise. So with that, I will go to the background here, representing the clinical actionable targets in intrahepatic cholangiocarcinoma. So the current state of genomic alterations in intrahepatic cholangiocarcinoma is here represented by more than 6,000 patients of targeted sequencing data indicating the main drivers of this disease, and some of them today being targetable, such as FGFR2 fusions, IDH, ERB2, or the HER2 amplifications, as well as using MSI high and tumor mutational burden high for immunotherapy. This is also interesting in line of multiple cancers, pan-cancers, based on the MSK impact data, where we can see that biliary tract cancers on the top part are among the cancer types with the highest targetability among most cancer types, with around 45% of all alterations having a potential target, whereas hepatocellular carcinoma, the other type of primary cancer in the liver, only has around 5% of targetability. Now, this is unfortunately is not leading necessarily to the fact that that biliary tract cancers are an easy disease to target. It would have been very nice if that was the case. And here, representing a couple of figures from the latest ENSCA consensus report from this year, publication from this year, actually showing that in the last decade or so, we actually have only seen an increased mortality rate of these cancers in most parts of the world, and primarily in the part of the world where we actually have implemented targeted sequencing or full genomic sequencing of these patients to identify potential targetability. So they should have really shown the opposite. So there is still a lot more in the equation that we don't know. One part of that potentially could be the increased knowledge of the risk factors that leads to this disease, where, yes, the part of risk factors that we have always described as a part leading to this disease, such as primary sclerosing cholangitis, is indeed highly associated with it, but the number of patients are much less than other larger risk factors, such as increased fat burden in the liver in line of MASLD, and also fibrotic states. Now, however, when we look at the timeline of systemic therapies in biliary tract cancer, both chemotherapy and targeted therapies, many trials since the relative success of the ABC02 in 2010 from the UK have actually failed. So that's the latter part between 2010 and 2020. Now, in the past 5 years, to also end the background on a high note, we have actually succeeded in quite a lot of improvements in line of treatment for these patients. So in 2020, we got pemigatinib approved in the US and later on also in Europe, but only in unresectable, locally advanced and metastatic tumors positive for the FGFR2 fusions, of course. And there are more drugs that have come online that also targets other FGFR2 alterations, I should say. This, however, is also a scientific achievement because this is one of the first patient stratification and biomarker-driven treatments in this area. Then we shortly after followed by approval of Tibsovo, which targeted the IDH mutation, and then again here in 2024-25 approval of the HER2 inhibitor zanidatamab, which is the drug that targets the ERB2 amplification that I showed you on the previous slide. We also have got approval of chemo and IO combination, but still in unresectable cases. With that, I will go to the first part of the talk, the genomic stratification of intrahepatic cholangiocarcinoma patients, looking at the perturbation mechanisms with novel therapeutic opportunities. So we did an integrated genomic characterization study a few years ago and stratified intrahepatic cholangiocarcinoma patients based on a multi-layered platform profiling approach, looking at mutations, transcriptome, structural variances, as well as epigenomic changes. We did this across a cohort in total of 496 intrahepatic cholangiocarcinoma patients. And on the left side, you see what is the were at that point a typical way of looking at patients. Basically, you have intrahepatic cholangiocarcinoma, and you have a median overall survival of around 12 months, which is also what was seen in 2010 in the ABC02 trial. Now we wanted to look at the potential drivers of intrahepatic cholangiocarcinoma, so subgrouping patient based on the IDH subtype, KRAS subtype, and P53 subtypes. And in this case of these three groups, none of those mutations were overlapping between each of the groups. Then we also have a fourth group called the undetermined group, which has also in other studies later on been termed as the wild type tumors. So the tumors that have none of those above driver mutation. They do not have IDH1/2 mutation, they do not have KRAS, they do not have P53 mutations. And then looked at the different platforms data in this, developing network vulnerabilities and matching drug sensitivities to these patients. Then looking at the oncoplot for these cohorts, both the discovery and the validation part, we see that there is already here at this stage a mutual exclusive grouping of these driver subgroups seen already in the oncoprint. And we see that the plot shows a clear separation of each of the mutant driver gene categories. This is clear in both the discovery as well as the validation cohorts that there is a marked mutational exclusivity. In fact, if we look at the spectrum of somatic alteration for each of the patient subsets, there is a marked enrichment of accompanied genetic alterations unique in each of the driver gene subsets. So if you look at the relatively boring plot here, which is a multi-Venn diagram for the 4 different groups based on IDH, KRAS, P53, and the undetermined, you see the size of the cohorts, subgroups within the cohort, but also the different passenger, if you will, mutations located within these groups that are still unique for each of the subsets, not only the driver mutation that the group was defined based on from the beginning. So these perturbations are recurrent and unique for each specific patient subset. And when we look beyond the lower penetrant passenger mutation, what we call the second-tier mutation based on their significance and penetrance in the disease, often these alterations are in epigenetic and DNA repair pathways, and considering some type of cooperative between them and the main prevalent driver mutations. Also, looking at the genetic substitution signatures, which is not shown on this slide, we see interestingly a distinct mutational signature emphasizing the specific genetic substitution patterns for each of these patient subsets. These patterns are masked when we look at across the full cohort, so across all the patients in the study. But when you enrich for the driver mutation, you actually start to see these interesting and more clean representations of perturbation mechanism for each of the subgroups. It's of course also reflective in the clinical demonstration and the clinical significance. Here seen by Kaplan-Meier analysis of both the time to recurrence as well as the overall survival in both the whole exome sequencing group, the targeted sequencing group, between discovery and validation cohorts. Now looking at the association between alterations and the pathways, the unique pathway association, the unique mutational signatures are also reflected in a specific functional pathway association. So there's a unique pattern that are seen in each of the layer of the data, here represented by one set, and it could be done or is done with the other sets as well, both in the mutational copy number alteration, transcriptomic data, and DNA methylation level of the data. When you look at the 4 different groups, the IDH group, KRAS, P53, and undetermined group, you see that the pathways, the perturbation mechanisms that are activated in these tumors are overall unique in each setting. So for example, for a P53 mutated group of patients, you see a clear association with Wnt signaling cell cycle, which is not so surprising, apoptosis, and of course P53 signaling itself, but also MAPK signaling. That overlaps a little bit with the KRAS group that has, again, other other unique perturbation functional pathway associations with the specific grouping of patients. Interestingly, the undetermined group that have neither IDH, KRAS, or p53 mutations, we see only one pathway association, one functional pathway association, and that's the mTOR signaling, which I'm asking you to remember to the next slide. So, in the next step, we took in this study, we tried to initially match cell lines in the lab, so cell lines in the dish, to these unique patient groups. So the first step of that is that they should, of course, have the driver mutation, but then using non-matrix factorization clustering of the whole exome sequencing data, they also have to match on as much as possible to the driver mutation, because those mutations are the ones that drive the perturbation mechanisms, the altered pathway association that I showed in the past slide. Then we tested a library of 525 drugs that are either FDA approved or in late stage clinical trial at the point of our study, and we try to determine the drug sensitivity score, DSS, as given in the slide. This is a method to establish the antiproliferative effect, the optimal dose, and the therapeutic window for each of these drugs. We also have a fifth group in this, as the chart you would have seen, called the mixed group. This is a mixed group is represented by a cell line which has more than one driver mutation, so it has both KRAS as well as an IDH mutation. And there, the reason for adding the mixed group is to ask the question: do we see an additive cumulative effect of the initial drug responses or not? So for the clean groups, matching the patients, the IDH, KRAS, p53, as well as the undetermined, in yellow you see the response to certain drugs, and those responses are unique for each of the cell lines compared to the matched response test in the other lines and the other groups. Now, in line of the undetermined, we again see a unique response, just like we had in the pathway associations, but in line of the mixed group, where there is more than one driver, we did not see an addition. So not an addition of the responses between, let's say, KRAS and IDH, but actually quite the opposite, that if you have more than one driver mutation, the entire response signature is destroyed. Do we sum the drug response to the biological processes? We see quite uniquely that each of the group of cell lines represented to the patient grouping IDH, KRAS, p53, undetermined, or the mixed, that we see unique responses to the drugs in the library that we tested. As an example, linking to the past slide where we saw mTOR pathway being the only associated functional pathway to an undetermined group of patients, we also only see mTOR inhibitors being effective on an undetermined associated cell line. This is, of course, data that needs to be tested if an undetermined associated patient, a patient that does not have IDH, KRAS, or p53, indeed would be a stronger candidate for an mTOR inhibitor, and that needs to be tested in the clinic, of course. However, one could envision, because in fact the FGFR2 fusion positive cases are in the undetermined is a subset of the undetermined group of patients, that a combination between FGFR2 fusions and mTOR inhibitors could be rational in the future, as we already start to see resistance to FGFR2 fusions in the FGFR2 fusion positive cases in the clinic. Now, have we actually a bit too much in the last decade been deeply focused on the mutational space, leading to, yes, those achievements that I said in the background on approved drugs particularly in the last 5 years? also exemplified by integrated genomics that are spearheaded by mutations, as I showed you in the part 1 of this presentation, but highlighting the fact that underneath the main driver mutations, the potential targetability of these patients, there is a large plethora of other changes that leads to specific oncogenic programs that are activated and potentially also could be targetable. So have we focused a bit too much on the mutational space, and should we actually consider looking into other areas of these tumors going forward, and not only relying on the mutational space? So, for example, looking at areas of in the change of the genome and the interplay of different cell types, looking at the pro-inflammatory signals, the microenvironmental clues, many of those functional readouts that are actually governed or controlled, if you will, by transcriptional changes. And this is an area in which we are coining a concept of exceptional responders that are not necessarily driven by a mutational space. So trying to understand precision chemotherapy by making what is actually by default imprecise, it's systemic chemotherapy targeting everywhere, to a more precise understanding. So regardless of many of the major achievements that we have discussed specifically in the last 5 years with approved drugs, chemotherapy still dominates this space in the clinic, and also presents therefore the rate-limiting step for optimizing treatment for these patients, as well as their clinical outcome. And the majority of the achievements that have been secured in the last years are actually more currently, maybe in the future it will be different, but currently in second line on unresectable cases, so locally advanced or metastatic cases, where we have targeted therapies as an option if you have IDH fusion, FGFR2 fusion positive, or a HER2 amplified or HER2 positive tumor. So we devised the first cohort of unresectable, so biopsy-based intrahepatic cholangiocarcinoma patients. Critically, these patients did not differ in their clinical characteristics, because that's quite often what is seen in the clinic. And we associate with the treatment outcome, such as age, stage, metastatic burden, or the ECOG performance score. There is no difference if you are in the latter part of the left side of the bell curve on the upper part in purple, or if you are, which we call rapid progressors, or in the green part on the right-hand side, long survivors. They also do not differ on most of the hematological and systemic markers. They only differ in two highlighted part, slightly above the 0.05 statistical line, and that is a rapid progressor, one that dies twice as fast as one would expect if you basically did nothing. And that is based on the ABC02 clinical trial, where a rapid progressor has a 6 months or less overall survival, compared to medium overall survival as seen in the ABC02 on 12 months, and the long survivors having twice as good a survival, so above 24 months compared to the median overall survival of 12 months. So in the rapid progressors, the only thing we see that is significant different in this cohort is that they have a slightly higher platelet count, and they have a slightly higher alkaline phosphatase ALP score for the liver. Otherwise, the overall clinical characteristics known to be associated with the treatment outcomes do not differ between rapid progressor RP patient or a long survivor LS patient. So patients are selected. We selected patient based on the median overall survival, the ABCO trial by Professor Valle and colleagues in UK, showing a median overall survival of 12 months. These patients we of course controlled or examined for by the CT imaging to confirm response or lack of response to the chemotherapy by radiology, so based on the RECIST percentage, RECIST score, as clinical stated by 30% reduction or more for the long survivors, or 30% increase or more for the rapid progressors. Innate chemotherapy is impacted by not only specific genetic or DNA-based alteration inside the cell. However, it is also affected by non-genetic alterations inside the tumor cell, as well as alterations in the microenvironmental cell, as I was indicated in the transition from part 1 to this part of the talk, which are typically non-genetic, exclusively non-genetic alterations. Therefore, an RNA-based profiling compared to a mutational profiling should provide more holistic view of these patients on their innate chemoresistance potential of a tumor, compared to actually devising the targeted sequencing of these patients. Excuse me. So we acquired the biopsies from this cohort of patients, and obtained. So we obtained the pre-treatment biopsies and performed digital histopathology analysis on the samples, and we overall found no differences in the total epithelial or tumor cell content of being a rapid progressor or a long-time survivor. So it's not based on the actual tumor cell content that these patients are responding differently to chemotherapy. However, in patients destined later on to become a rapid progressor, we identify higher stromal and lower immune cell infiltration, suggesting a link between these microenvironmental cells and the chemotherapeutic outcome. And we did this in a multi-regional sampling approach, shown in the lower part of this quadrant, looking at both the non-malignant regions of the biopsies, the invasive front of the biopsies, the tumor core of the biopsies, as well as the tumor stromal part of these biopsies, and all data being profiled by transcriptomic analysis. What I will be showing you in the rest of the talk is based on the tumor core, but we did the same for all the areas. So in this study, we used an approach called TempoSeq, a methodology called TempoSeq, where you can scrape off part of the histology slide and send for profiling. We devised a rapid progressor, long-time survivor, and RPLS signature, which is consistent of 504 differentially expressed genes, and this is how the heat map looks when you cluster the patients. In the next part of the study and the next slides, to go between different data types, as well as different platforms for transcriptomic profiling, sequencing arrays, Illumina sequencing versus Tempo sequencing, and so forth. And also to go between different cohorts, we collapsed the signature of genes into an RPLS score based on this relatively simple matrix. So to determine the generalizability of the RPLS signature and validate, as well as define the signature in early stage disease now, instead of the unresectable late stage disease or metastatic disease on the biopsies, we evaluated its prognostic potential or performance in 644 resected patients represented on this slide from 5 international cohorts, including my own cohort and 4 other cohorts that we could get our hands on. As you can see, higher RPLS score from the surgical material was associated with significant decreased 5-year survival. And these data indicate that the molecular basis of chemosensitivity established early on, not necessarily late on, as one could have envisioned with the biopsy data during cholangiocarcinogenesis. Next, we aim to determine its cellular origin of the RPLS signature genes. To do this, we applied an approach called digital cytometry. This involved constructing gene expression signatures that can differentiate different cell types in single cell sequencing data from intrahepatic cholangiocarcinoma. Then applying these signatures to our bulk biopsy transcriptome data from the biopsies to infer cell types that drive the expression changes of the individual genes. And then also, of course, remembering that the data that I'm showing now is from the tumor core part of the data, not the tumor stroma, not the normal areas, as well as not the invasive front area. To cut a long story short, we found that tumor and myeloid cells were the dominant contributors to the RPLS signature genes, reflecting heterotropic communication between these two cellular compartments, tumor and tumor microenvironment. By isolating tumor-infiltrating myeloid cells, so the CD68 positive cells shown in magenta, from the same biopsies, we were able to experimentally verify several of the digital cytometry predictions, the genes that are shown in the gray dots, as well as specific genes we were profiling for using GeoMax digital spatial profiling, revealing a significant immune dysfunction of the rapid progressor-associated myeloid cells compared to those found in long-time survivors. So the rapid progressor biopsies, the rapid progressor patients, the rapid progressor tumors have a dysfunctional myeloid compartment compared to the long-time survivors. And this is associated with the tumor, to the response to chemotherapy. Now, using modeling of single-cell RNA data, using transcription factor activity analysis, cytokine activity analysis, as well as ligand receptor interaction, conferring ligand receptor interactions, subtracting what is similar between the rapid progressors and the long-time survivors, leaving only on the screen what is unique for a rapid progressor, so therefore the rapid progressor view of this data. We looked at the communication between CAFs and rapid progressor-like tumor cells, and rapid progressor-like tumor cells and myeloid cells and T cells. As you can see, there is very limited communication between CAFs, T cells to the rapid progressor-like tumor cells, as well as in the other direction, but a very, very high proportion of communication going bidirectional between rapid progressor-like tumor cells, as well as the myeloid cell compartment. And interestingly, that communication bidirectional is also represented by interleukin-6 molecule that I would like you to remember, and that I will come back to shortly. Now, CCA can arise within the liver, as we have been discussing so far, so intrahepatic cholangiocarcinoma, but it can, of course, also, as you likely all know, arise outside, so extrahepatic cholangiocarcinoma, perihilar in the bifurcation, as well as distal cholangiocarcinoma. So we first show in our smaller cohort of perihilar cases, or in the bifurcation of the common bile duct, that the RPLS signature does not distinguish survival or response of these patients based on the RPLS signature, which is in strict difference to all the data I showed you for 644 cases of intrahepatic cholangiocarcinoma. Now, this is a small cohort. There is only one larger cohort of resected extrahepatic cholangiocarcinoma. This is likely a mix between both perihilar and distal. We actually don't know. On the Montal et al. Cohort, it's all distinguished as extrahepatic cholangiocarcinoma, so representing these two cases. And this was in collaboration with Professor Joseph Lovett's team in Mount Sinai in New York. Here we tested then in 182 cases that they have in their data, the survival association of the RPLS signature with the ECCA. And despite a continued robust prognostic performance in intrahepatic cholangiocarcinoma, the RPLS signature failed to differentiate outcome in patients with the ECCA, also in this larger cohort. So it seems to be quite unique in line of intrahepatic cholangiocarcinoma. These data then suggest that the liver provides a unique microenvironment that engages distinct oncogenic programs, and those oncogenic programs are predicted by the RPLS signature genes or score, significantly impacting patients' benefit from chemotherapy. So that is the RPLS signatures present an important association with the liver microenvironment. To start homing in on that, we looked at metastatic liver metastatic cases that we can get our hand on, or data we could get our hand on, and the biological process. So the biological process represented by the RPLS signature genes are unique, associated with only liver metastasis in this data. These data are in more than 1,000 patients, pan-cancer metastasis from the MET 500 and the POC 570 cohorts. And the RPLS signature only predicts liver metastasis, not metastasis in any of the other organs or anatomical sites. So as stated previously, the liver provides an environment that are important for the response, which is related to the myeloid dysfunctional that I presented earlier in the biopsies for the RP, the rapid progressor cases. Excuse me. Now, this does not really directly argue that the liver is indeed presenting a different environment, and that environment is responsible for the response to chemotherapy, longitudinally speaking. So in the outcome data, for that we need a very unique cohort available where we have primary tumor site outside the liver. We have metastasis to the liver from that primary site. We have a cohort of patients from which transcriptomic data has been performed, and we have outcome data of these patients followed up over time following chemotherapy, and that combination of requests or requirements is very difficult to find. There is, to my knowledge, only one such complex data set, and that's is the ESCORT clinical trial team in UK on the new EPOCH trial. So to fully define the statement that the liver provides a unique environment, and that unique environment related to the myeloid dysfunctionality of the rapid progressor patients is the cause of the response to chemotherapy. We used this cohort from the ESCORT and collaborated with the ESCORT clinical trials team in UK and tested the RPLS signature in their primary tumors. They resected liver metastasis of these patients with colorectal cancer in the liver, metastasized to the liver, treated with chemotherapy, pre and post liver resection chemotherapy. When quantified in the primary colorectal tumor tissues, the RPLS signature failed to predict patient outcome as we would have predicted. However, when we look at and quantify the RPLS signature in the liver metastasis, and predicting its progression and predicting the outcome based on progression-free survival or overall survival. We see in this and in this part on a new APOC trial that the RPLS signature, again, just like in line of intrahepatic cholangiocarcinoma, are able to distinguish outcome of these patients. So evidently, the RPLS signature has a potential to predict chemotherapy outcome when evaluated in liver lesions independent of the primary tumor diagnosis. Also, correlating the clinical features of and with the RPLS score. The RPLS score is the statistical most powerful variable in this trial data on predicting patients' outcome in a new APOC trial. Now going back to the IL-6 part that I showed you in the cell-cell communication data, we some time ago predicted the role of serum IL-6. So we know that BTC or biliary tract cancers are characterized by a dense dysplasia as part of the tumor microenvironment. Here, chronic inflammation plays a crucial role in promoting the cancer by the interaction between the cancer epithelial as well as the stromal compartments, and that such crosstalk is coordinated by pro-inflammatory factors, and IL-6 is one of those factors that plays a cardinal role. This has also before been mentioned in the ABC02-3 trial data showing an elevated IL-6, serum IL-6 with the BTC diagnosis, associated with the BTC diagnosis. So we looked at 452 biliary tract cancers from 3 different Danish hospitals enrolled between 2004 and 2017. Samples were obtained and stored according to remarks of biomarker analysis. Patients all have advanced, so similar to the biopsy part of the study where the RPLS signature was devised, of locally advanced and metastatic disease. We analyzed a total of 1,590 serum samples for 3 different markers, IL-6 of course, CA19-9, that is the current standard marker for diagnosis and prognostic. Is not a very good marker, but still it's the standard one, as well as YKL40 at the time of diagnosis, so the pretreatment samples, as well as continuous variables until prognosis of this disease, so longitudinally following these patients. YKL40, for those that do not know, YKL40 is a chitinase 3-like 1 glycoprotein that is within the IL-6 pathway. So on the left part, we see as one short representation of this study, the ability of these markers to predict death at 1 year, performed by the ROC curve analysis, showing in the red and blue IL-6 and YKL40 being statistically more significant than CA19-9, which is more alike a flip of a coin, so close to the middle 0.5 line. So on the right hand side, we look at the post-hoc analysis of this data and the ability of these markers to predict survival for inclusion as well as longitudinal across all time points up to 3 years, which is a long time follow-up of these patients when they have a median overall survival of 12 months, according to the ABC02 trial, as presented a few times. And then suggesting that the pretreatment CA19, this data suggests that the pretreatment CA19-9 is best in the longer times of crossing at the 1.5-year line following the median overall survival, whereas in the first 1.5 years of a patient following diagnosis, IL-6 is a really, really strong diagnostic marker to consider. And that is one of the markers that we saw in the communication for the rapid progressors. Now, this is IL-6 data in circulation, so it's serum IL-6. So what about IL-6 in the tumor tumor context? Here we are showing a really new study that's just been accepted yesterday in Gut, so you can read it very soon, I hope, where we use Visium HD, so true single-cell spatial transcriptomics on 14 ICC tissues, looking at all the representation of cell types, and then superimposing IL-6 expressing cells on these to see if indeed, as expected, tumor cells themselves, not only the myeloid compartment, indeed is expressing IL-6 as well, provoked by the role of chemotherapy on these patients. And this is indeed what we see. We have IL-6 high patients as well as IL-6 low patients, and that there is a direct association between the IL-6 high patients, where we see a unique activation of the epithelial to mesenchymal transition. We see a signaling between the cancer cells with high IL-6 expressing and their neighboring immune cells, which demonstrates that the tumor-specific IL-6 in that context of the tissue now actively reshapes the tumor microenvironment, among others, including the stromal interaction, so the CAF interaction in these patients. Now, summarizing and concluding on our RPLs model and the data that I've shown you, and parts of data that I also did not have time to show you. In an intrahepatic cholangiocarcinoma associated with a future long-time survival phenotype, tumor cells may harbor genetic insults such as IDH mutations. We already know that. These lead to tumor cell behavioral states defined by a high expression of TCA cycle, and cellular adhesion molecules, as well as tumor cell proliferation. In coordination, myeloid cells have increased anti-tumor functionality and decreased antigen presentation, while there is a high proportion of effector T cells related to Tregs. Collectively, these cellular ecosystems contribute to a hepatic microenvironment with proficient anti-tumor immunity, which, following chemotherapy, fosters initial tumor sensitivity and active immunogenic elimination, culminating in what we call the long-time survival or the exceptional responder patients. In contrast, on the right-hand side, we have ICCs, or intrahepatic cholangiocarcinoma, destined to become rapid progressors. These are tumors with tumor cells harboring genetic insults such as KRAS and TP53 mutations. These lead to tumor cell behavioral states defined by high glycolysis, expression of checkpoint molecules, fast tumor cell proliferation. There is an increased abundance of plasmatoid dendritic cells and antigen presentation of myeloid cells in general. The myeloid cells are dysfunctional, as I showed you, as well as a high proportion of Tregs and lower abundance of effector T cells. Collectively, these cellular ecosystems contribute to hepatic microenvironment in a state of immunological inertness, defined by immunosuppressive molecules and cells culminating in tumor tolerogenicity. Following chemotherapy, these tumors exhibit a higher degree of intrinsic chemoresistance and additional fail to undergo immune-mediated elimination, resulting in a rapid progressor patients, so our RP patients. In red are the patients, in red are the people in the lab that contributed to the studies that I've shown you. This is the full current lab. So Komaruk have led all these studies. Orestes Noises performed the spatial transcriptomics. Virak Gill, a very talented PhD student who has just finished and just got her paper accepted in GOT, led the IL-6 study and the initial IL-6 study on serum level, and IL-6 as a potential biomarker to follow patients' response to chemotherapy was led by Dan Høgdahl from the clinic here in Denmark. These are some of the collaborators on the RPLS signature. Thank you very much for your attention and for listening to this talk.
Thank you, Professor Andersen.